Hello friends, we will now start the topic DNA replication in a series of lectures which will include the process of replication involving initiation, elongation, termination, the enzymes and accessory proteins involved in replication, and the regulation of replication. In today's lecture, we will revise the structure of DNA and then move on to talk about the semi-conservative nature of DNA replication and the experiment to prove it. DNA replication is the process through which the genetic information is passed from parent to progeny. Cells need to make a copy of DNA before dividing so that each daughter cell has a complete copy of genetic information as can be seen on your screen. Replication should be foolproof and errors should not occur. Even if errors occur, they should be corrected in the time. Replication takes place in the prokaryotic cell along with transcription and translation in the cytoplasm. Prokaryotic DNA is not present in a well-defined nucleus. Instead, it is present as a nucleoid in the cytoplasm of prokaryotes. Before exploring the process of DNA replication, let us first recollect some basic structural aspects of DNA. We all know that DNA is a double-stranded helix made up of two complementary chains of nucleotides. This is the 5' end of one strand. This is the 3' end of the strand. Here we have the 3' end of the other strand and the 5' end of that strand, which shows the antiparallel nature of DNA. Each strand serves as a template during replication guided by specific base pairing rules. Adenine base pairs with thymine and cytosine base pairs with guanine. A purine always base pairs with a pyrimidine. You can see these hydrogen bonds between the nitrogenous bases, and we can also see here the covalent phosphodiester bonds between the sugar and the phosphate. Thus, you have a sugar-phosphate backbone running antiparallel in nature to which the nitrogenous bases are attached. Let us have a structural overview of DNA replication. DNA replication relies on the complementarity of the DNA strands. It follows the ATGC rule or the Chargaff's rule. The process can be summarized as the two DNA strands will first of all separate so that each of them acts as a template for the synthesis of new strands. The two newly made strands are called the daughter strands and the original strands are called the parental strands. This is a double-stranded DNA molecule. The first step is the separation of the parental duplex to form two single-stranded DNA template molecules. Each of them will act as a template, and to this the complementary nucleotides would be added so as to give rise to two completely formed daughter duplexes, which are actually exact replicas or copies of the parental duplex. In short, this is a parental duplex. The two strands have separated and the two newly synthesized strands are being formed here, one in the form of leading strand, the other in the form of lagging strand, where the incoming nucleotides are added on the basis of the complementarity, and the product of this replication is the formation of two daughter duplexes which are actually identical replicas or copies of the parental duplex. So this is a parental duplex where just keep a note that both the strands are red in color. After replication, two completely formed daughter duplexes are formed And just remember and just keep in mind that the daughter duplexes have one strand red and the other strand yellow. We'll be talking about this in the lecture soon. When replication was being studied, three possible replication models were proposed or hypothesized. The first one was semi-conservative replication, the second was conservative replication, and the third was the dispersive mode of replication. Let us first talk about the semi-conservative model of replication. This model, which was later experimentally confirmed, proposes that each of the two parental DNA strands serves as the template for the synthesis of new complementary strands. As a result, each daughter DNA duplex consists of one parental DNA strand, which here is blue in color, and one newly synthesized strand, which is red in color. This mechanism ensures that half of the original DNA is conserved in each daughter duplex, and hence the name semi-conservative. The second is the conservative model of replication. In this model of replication, the entire parental DNA molecule is conserved as a single unit. After replication, one daughter DNA duplex would consist of both original DNA strands, while the other would consist of two newly synthesized strands. This means the original DNA double helix remains completely unchanged and is passed along to one daughter cell, while the other fully new DNA duplex goes to the other. The third model is the dispersive model of replication. According to the dispersive model of replication, the parental DNA is broken into segments and both daughter DNA duplexes contain fragments of old and new DNA. Each strand in the daughter DNA would be a patchwork of parental and newly synthesized segments. This model implies that after each round of replication, the DNA becomes more hybridized and less distinguishable between old and new DNA sequences. As you can see on your screen, the daughter duplexes in the dispersive mode of replication are made up of bits and pieces of red and blue DNA. In 1958, Matthew Meselson and Franklin Stahl devised a method to investigate these models. They wanted to distinguish between the parental strand and the daughter strands. For example, I used two different colors to differentiate the parental strand and the daughter strands. They found a way to experimentally distinguish these two strands. What they did was they used the isotopes of nitrogen and density gradient centrifugation to prove the hypothesis. Let us try to understand what exactly they did. Supposing we take E. coli as our bacterial culture and grow it in a medium containing the heavy isotope of nitrogen, that is N15 nitrogen. Allow this E. coli to grow and after 24 hours, isolate the DNA and carry out density gradient centrifugation. The DNA will settle at a density of say 1.72. Supposing we have another flask containing which contain the media containing the light isotope of nitrogen, that is N14, and allowed E. coli to grow in this flask. After 24 hours, if we isolate the DNA and carry out density gradient centrifugation, the DNA will settle at a density of 1.71. This was actually exploited by Meselson and Stahl to prove the semi-conservative nature of DNA, as we will see in the coming slides. Their experiment can be summarized like this. Grow E. coli in the presence of the heavy isotope of nitrogen for many generations. All the DNA now would be heavily labeled and we will call it the heavy DNA. Switch the E. coli from this heavily labeled medium to a medium now containing the light isotope of nitrogen, that is the N14 nitrogen. Allow the cells to grow and then collect the sample of DNA after different time intervals and analyze the density of the DNA by centrifugation using cesium chloride density gradient centrifugation. Let us try to understand this. So grow E. coli in the medium containing the heavy isotope of nitrogen. Allow it to grow for several generations, as a result of which the entire DNA is heavily labeled. Now from this flask, transfer the culture into a medium containing the light isotope of nitrogen. Allow the cells to grow for 20 minutes and isolate the DNA and centrifuge it. This DNA is that of the first generation of the E. coli cells. From this same flask, now isolate the DNA after 40 minutes. This would give you the second generation DNA. So as per the experiment, if DNA replication is semi-conservative in nature, then the parental DNA here, the parental duplex, is all heavily labeled DNA, and hence after density gradient centrifugation, it should give us one single band here coinciding with the heavy DNA. After 20 minutes, that is after the first cycle of replication, the two daughter duplexes here are such that both of them contain one parental strand and the other newly synthesized strand. Mind well, the parental DNA here is N15 DNA and the daughter DNA here is N14 DNA. So if we take these DNA duplexes now and carry out density gradient centrifugation, you should be getting one single band of that of the hybrid DNA, which coincides with the densities of N14 and N15 DNA, which is intermediate between N14 and N15 DNA. If DNA replication is conservative, then the parental duplex here again is heavily labeled, so it should be giving you one single band of N15 DNA. But after one cycle of replication, mind well, if DNA replication is semi-conservative, after one cycle of replication, you will be having two daughter duplexes. Just look at the duplexes here. One daughter duplex is completely conserved and it is made up of the old DNA, that is the heavy DNA, and the other duplex is completely newly made DNA, which is made up of the N14 DNA. This DNA molecules after density gradient centrifugation should be giving you two distinct bands. One band which coincides with the heavy isotope of nitrogen and the other band which coincides with that of the light DNA. Let us move on to the third model. If at all DNA replication is dispersive, then the parental duplex here, like we have discussed just now, would be giving you a single band coinciding with the N15 DNA. But after one cycle of replication, after 20 minutes, both the daughter duplexes are made up of bits and pieces of old and new DNA, that is heavy DNA and light DNA. This DNA after density gradient centrifugation should be giving you a single band coinciding with a density which is intermediate between N14 and N15 DNA. So we have seen the three models and their predicted results. As per that, the conservative model, we have already seen that after the first round of replication should be giving us two distinct bands, one coinciding with the heavy DNA and the other coinciding with the light DNA. But every time the experiment was carried out by Meselson and Stahl, it was found that after the first cycle of replication, it led the DNA led to the formation of only one single band with a hybrid density. This completely ruled out the conservative model. Now, the two models that were left for Meselson and Stahl were the semi-conservative model and the dispersive model. As per that, the semi-conservative model tells us that the parental DNA, which is made up of the heavy isotope of nitrogen, would be giving us one single band coinciding with the heavy DNA. After the first cycle of replication, semi-conservative model proposes that it should be giving us one distinct band coinciding with the hybrid density. After the second round of replication, that is after 40 minutes, we have this 4 daughter DNA duplexes here. As you can see here, out of these 4 daughter duplexes, 2 DNA duplexes are made up of completely new DNA, that is the light DNA. And 2 daughter duplexes are made up of hybrid DNA. This on density gradient centrifugation should be giving us 2 distinct bands, one of that of the hybrid DNA and the other of that of the light DNA. Whereas if the DNA replication is dispersive, then after the second round of replication, as you can see, all the 4 daughter duplexes here are made up of a mixture of heavy and light DNA. This should be giving us one single hybrid band coinciding with the density intermediate to that of N14 and N15 DNA. However, this band would not be a sharp band as this, but it would be a dispersed or a slightly broad band. When this experiment was carried out by Meselson and Stahl, it was found out that the second generation DNA every time gave them 2 distinct bands, one of the hybrid DNA and other of the light isotope of DNA. This should have completely ruled out the dispersive model of replication. So, as per the predictions, the first generation DNA would always give one single band, as a result of which the conservative model is ruled out. And after the second generation, it should be giving us 2 distinct bands like that of the semi-conservative model. This should have ruled out dispersive model of replication. But then there was a group of scientists who always believed that dispersive is a random method of replication, and after the second generation, it was quite possible that it would lead to two distinct bands. Hence, to prove this fact, Meselson and Stahl took the first generation DNA. So they took the first generation DNA molecules and tried to distinguish the content of DNA between the semi-conservative and the dispersive models of replication. So as we have already seen, the parental DNA is the heavy isotope of DNA, that is the heavy DNA. And after the first generation, you will have 2 daughter duplexes such that one strand is heavy DNA and the other strand is light DNA. If this DNA is taken, the first generation DNA is taken, and if it is subjected to denaturation, then you should be getting 4 single-stranded DNA molecules such that 2 of the strands here are heavy DNA and 2 of the strands are light DNA. This on density gradient centrifugation should be giving you 2 distinct bands, one coinciding with the heavy DNA and the other coinciding with the light DNA. If DNA replication is dispersive, then the first generation DNA would be a mixture of heavy and light DNA, and hence would be having a density which is hybrid of that of the N15 and N14 DNA. So if this first generation DNA molecules are taken and they are subjected to denaturation, then it would result into 4 single-stranded DNA molecules, each of one which is made up of bits and pieces of N14 and N15 DNA. These four single-stranded DNA molecules, if subjected to density gradient centrifugation, should be giving us one single hybrid band of DNA. Now, when Meselson and Stahl actually carried out this experiment, every time they carried out this experiment, the first generation DNA molecules after denaturation and centrifugation gave them two distinct bands of DNA, reconfirming that DNA replication is semi-conservative in nature. This lack of the hybrid DNA strands allowed Meselson and Stahl to rule out the dispersive model of DNA replication. With this, we come to an end of this lecture where we have discussed the first characteristic of DNA replication, and that is the DNA replication is semi-conservative in nature. This tells us that half of the parental DNA molecule is conserved in each new double helix paired with a newly synthesized complementary strand. We have seen how Meselson and Stahl have proved the semi-conservative nature of DNA replication. This feature of DNA replication is a universal feature, which means that it is obeyed by all living cells. In the coming lectures, we will talk about the other characteristics of DNA replication, particularly the semi-discontinuous nature and the bidirectional nature of DNA. Thank you.